Hello? Okay, gang, can everyone hear me?
Yes.
Excellent. And do the slides move?
Yep.
They do. Okay. And it's time to begin. So let's have a word of prayer. Father, thank you for this day, cold though it may be. Thank you for the chance to learn, even though it's not under ideal conditions. Give us ears to hear, eyes to see. Help us learn of our bodies, learn of the Creator who made us, learn of your grace that will one day remake us when you call us from the earth on that day. In Jesus' name, amen. All right. So we talked before about the transport of oxygen through the blood. Typically, oxygen is poorly soluble in an aqueous solution. You can see the gee whiz number here. There's, even with a partial pressure of oxygen, you really don't have much oxygen dissolved per 100 milliliters of blood. So per deciliter of blood, you just don't have that much dissolved because not much dissolves in an aqueous solution. So what do you need to up that? You need to provide a carrier. And that carrier is the protein hemoglobin. So hemoglobin is found in blood cells. Red blood cells are packed to the gills with it. That hemoglobin will carry the lion's share of the oxygen. Consequently, if your red blood cell hemoglobin concentration are normal, you're able to carry far more molecular oxygen than you are if we only depended upon the solubility of oxygen in the aqueous plasma. So 20 milliliters per deciliter, I think you'll agree, is quite a bit more than 0.3 milliliters per deciliter. That's how much you carry when hemoglobin numbers are normal with respect to your blood. So the hemoglobin carries the oxygen. It is encased inside red blood cells. The hemoglobin is a protein. It consists of 4 polypeptide chains. 2 of those chains are the same. 2 of those chains are alpha globins. The other 2 polypeptides are beta globins. And then each globin has a heme molecule. And the heme contains essential iron. And that iron is the business end, if you will, of hemoglobin. That's the piece that actually carries the oxygen in the hemoglobin. So here is a hemoglobin molecule. So in the light blue, you have the beta chain. Each beta chain has a heme, as you can see in the red. Below it, you have the purple colored polypeptides. Those are alpha globin chains. Hemoglobin has 2 beta chains, 2 alpha chains. Each of the globin chains has its own heme group. If we look at the structure of a heme, it's a tetraperol molecule. This tetraperol contains these nitrogens. These nitrogens form div bonds to an iron, and that iron is set very stably within the heme molecule. The hemoglobin then is going to bind, rather the heme in the hemoglobin will bind to the oxygen essentially in a manner perpendicular to the plane of the heme. The heme is more or less perpendicular. The iron is, for our intents and purposes, within the plane of the heme. That molecular oxygen is going to bind perpendicular to the plane of the heme. So the hemoglobin can consist either bound to oxygen, which is called oxyhemoglobin. It can be bound, unbound to oxygen, which is called deoxyhemoglobin. We'll see this is not a hard and fast rule. Hemoglobin can be sort of bound by oxygen and not completely with oxygen. When hemoglobin is bound by oxygen, there's a color shift. That color shift is detectable, and this is the basis of a pulse oximetry electrode that is placed on your finger. This pulse oximetry electrode shines a light through the center of your finger, and based on that color shift that occurs in hemoglobin when it's bound to oxygen, it will be able to detect the oxygen saturation of your blood. That's how a pulse oximetry electrode works. If the hemoglobin ends up getting oxidized, that's called methemoglobin. And methemoglobin has a green color. That's how you tell the difference between unoxidized hemoglobin that's still functional, it's red, and oxidized hemoglobin, it's green, that's methemoglobin. If the hemoglobin gets bound by carbon monoxide, then it becomes carboxyhemoglobin. Carbon monoxide has a much higher affinity for hemoglobin than does molecular oxygen. So molecular oxygen can be severely outcompeted by carbon monoxide. And if you bind carbon monoxide to your hemoglobin, you need to remove yourself from that carbon monoxide very, very quickly, or most of your hemoglobin will get bound up by carbon monoxide, and your hemoglobin will be taken out of action. You will be unable to breathe, essentially. Now, if your red blood cell count is low, then you suffer from a condition called anemia. And anemia will decrease the oxygen carrying capacities of the blood, because the lion's share of that oxygen that is carried in the blood is carried by hemoglobin. If your red blood cell count is low and you have anemia, that means your oxygen carrying capacity of your blood is abnormally low. If your red blood cell count is low, then your kidneys will make erythropoietin. That will jack up red blood cell production by the bone marrow. And androgens also increase blood cell production by the hemoglobin. Therefore, males will tend to have slightly higher hematocrit than females because of those androgens. Hemoglobin has a form in which you find it, and that form is going to vary depending upon how much oxygen is bound to it. So let's go from here to our makeshift chalkboard. Yeah, that's not working. Let's try that again. There we go. All right, so and you look at the total let's try something that actually writes. Total hemoglobin in the body, you have hemoglobin that is bound to oxygen, and hemoglobin that's not bound to oxygen. Now, when hemoglobin is bound to oxygen, it is in a conformation that's called the R state. The R state. The R state has a very high affinity for oxygen. When hemoglobin is not bound by oxygen, it's in the T state. And this has a low affinity for molecular oxygen. And hemoglobin is going to toggle between these two states based on the partial pressure of oxygen. So at high partial pressures of oxygen, you favor the R state. Hemoglobin will tend to take up oxygen. At low partial pressures of oxygen, you will favor the T state. Hemoglobin will tend to give up oxygen. So we have, when it comes to hemoglobin, the so-called loading and unloading reaction. And these are linked to the transition from the state to the T state. The loading reaction refers to the loading of hemoglobin with oxygen. This occurs when hemoglobin is in R state. It occurs when hemoglobin, therefore, is in the lung, because the lung turns out to be area of your body where the partial pressure of oxygen is high. The unloading reaction refers to the release of oxygen by hemoglobin, and that is typically when hemoglobin is in the T state. Hemoglobin will be in the T state when it is in an area of low partial pressure of oxygen. That tends to be in the tissues and in the venous vasculature. So in the lungs and in the arterial vasculature, hemoglobin will largely be bound with oxygen. In the tissues, in the capillaries, and in the venous blood, hemoglobin will largely be in the T state and will largely be devoid of oxygen, or will not be fully loaded with oxygen. And this is illustrated very nicely in this binding curve, this hemoglobin oxygen dissociation curve. In arterial blood, where the partial pressure of oxygen is about 100 millimeters of mercury, hemoglobin can get fully saturated with oxygen. The reason for that is very simply that as hemoglobin transitions to the R state, it binds oxygen very, very easily. So once you start binding oxygen, you bind those first few oxygens, and the next oxygens you bind much more easily. In the venous capillaries of the tissues, the partial pressure of oxygen is only about 40 millimeters of mercury. Here, hemoglobin is only about 75% saturated with oxygen. That's a quarter of oxygen that's bounded to the hemoglobin. Excuse me, that's a quarter of the oxygen that's bound to the hemoglobin gets dropped off, it gets unloaded in the tissues. That's due to the fact that as hemoglobin transitions to the T state in areas of low partial pressure of oxygen, hemoglobin will give up oxygen much more easily. And this transition between R and T state is influenced by the partial pressure of oxygen. Now, there's other factors also influence the loading and unload of oxygen by hemoglobin, and those have to do with the carbon dioxide and pH of the blood. So let's go once again back to our makeshift blackboard. So in the tissues, we have lots of cellular respiration. Cellular respiration is going to consume oxygen, and it's going to produce carbon dioxide. The other thing that cellular respiration will produce is it will produce hydrogen ions. So it produces acid. Now, hemoglobin when it transitions from the R state to the T state, hemoglobin is actually capable in the T state binding carbon dioxide. It binds to the amino terminus of some of the globin polypeptide chains, and some of the side amino acid chains are also able to bind hydrogen ions too. When hemoglobin binds carbon dioxide and hydrogen ion, that stabilizes the T state. By stabilizing the T state, you take hemoglobin and you make sure that it stays in the T state while it's in the tissues. That way the tissues can take up as much oxygen as they require. So this is going to facilitate the unloading reaction. Then when it goes back to the lungs, the hemoglobin is going to transition to the R state, and it's going to give carbon dioxide, and it's also going to give up hydrogen ions. What does it do with those? We'll see what it does with those in just a bit. Let me pause right here, see if this part is clear. Ask any questions now, please. Very good. Then we will go back to our hemoglobin story here. The blood cells in the tissues possess a enzyme called carbonic anhydrase. Carbonic anhydrase catalyzes this very reaction right here. The conjunction of carbon dioxide and water to form carbonic acid. Carbonic acid then disjoins spontaneously to form hydrogen ion, which is bound to hemoglobin and bicarbonate. Bicarbonate is then pumped to the outside, and chloride is pumped to the inside of the red blood cell. This is to a bicarbonate chloride antiporter. Bicarbonate chloride antiporter. And it works this way because bicarbonate concentrations are very high inside the red blood cell, and outside the red blood cell, chloride concentrations are very high. Then, when the red blood cell gets to the lungs, we now have the hydrogen ion being released. It is therefore joined to bicarbonate. Bicarbonate is now brought into the red blood cell because bicarbonate levels are high in the lung, inside the cell. Chloride is then pumped out of the cell in exchange for bicarbonate going into the cell. So this process that essentially worked in the opposite direction in the tissues now changes direction in the lung. This is called the chloride shift. the chloride shift. Now that hydrogen ion is taken up and it makes carbonic acid. Carbonic acid is then disjoined by carbonic anhydrase to carbon dioxide and water, and the carbon dioxide is then exhaled into the alveolus, which is then released when we exhale. The CO2 that is bound to the T state hemoglobin is released when hemoglobin transitions to the R state. This CO2 is then also released and is exhaled as well. So we see this process is reversed essentially. What occurred in the tissues undergoes reversal, and that's driven by the transition of hemoglobin from the T state to the R state in the lung. Temperature, I might add, also increases the release of oxygen by hemoglobin. Higher temperatures affects the bond between the iron and oxygen, and that favors the T state of hemoglobin. During exercise, increased activity will cause muscles to generate increased heat. Even a rise of a few degrees in temperature will cause more loading of oxygen, and this helps the increased metabolic demands of the muscles. And you can see during mild exercise, 80% of the hemoglobin in the venous blood can return as deoxyhemoglobin. During heavy exercise, almost half of the hemoglobin returns as deoxyhemoglobin. And even at altitudes of 10,000 feet, the percentage of hemoglobin that's bound to oxygen during aeration is still quite high, 9%. And this shows you essentially the exchange essentially of hydrogen ions and bicarbonate in the tissues and then reversing its movement in the lungs and so on and so forth. So bicarbonate is going to serve as a buffer reserve, and the increased acid that you produce and getting buffered by bicarbonate. That's why the pH of your blood will still stay around about 7.4. So if somebody is trying to sell you alkalinized water because it'll help alkalinize your blood, it's a put on, it's a scam, don't buy it. You have many buffer systems in your bloodstream, you don't need it. So this, this curve shows that hemoglobin essentially has a so-called positive cooperativity. Positive cooperativity, meaning that as the hemoglobin binds oxygen, it gets better and better at it, and then in lower partial pressure of oxygen, as it releases oxygen, then it gets better and better at releasing it. The sigmoid curve of the hemoglobin oxygen dissociation illustrates this very nicely. Now, in increased acid levels, the oxygen binding of hemoglobin decreases. This is going to increase your release of oxygen in heavily exercising acidic tissues. And this is called the Bohr effect. And this is due to the fact that the acid is greatly stabilizing the T state and keeping hemoglobin in the T state and making sure that it will never snap back to the R state, even when hemoglobin is giving up lots and lots of oxygen. Now, additionally, in red blood cells, there's a metabolite called 2,3-diphosphoglycerate. 2,3-diphosphoglycerate. It's going to bind to the central cavity of hemoglobin that's formed during the T state. This cavity is very positively charged. This negatively charged 2,3-diphosphoglycerate, otherwise known as 2,3-DPG, greatly stabilizes the state, helps hemoglobin release its oxygen during circulation through the tissues. And this is also a metabolite that is put into blood that you donate to make sure that when the blood is given, it will essentially release whatever oxygen it has to the tissue in which it finds itself once it's given back into a human body. This dissociation graph at different pHs shows you the Bohr effect. It will decrease the affinity of hemoglobin for oxygen and guarantee more efficient delivery to oxygen, of oxygen to tissues during periods of acidosis. This also means during periods of pathologic acidosis, like for example, if you have uncontrolled diabetic ketoacidosis, or if you have some other acidosis due to aspirin poisoning or something like that, you will tend to have decreased loading of oxygen, of hemoglobin by oxygen in the lungs, and that will decrease delivery potentially to the tissues because you have oxygen you're bringing to it. And this is called the Bohr effect, the Bohr effect. That's why acidosis, one of the signs of acidosis is panting, in order to try to get you to deliver more oxygen and also to get rid of excess acid. That's one of the ways your body can get rid of excess acid, because you unload that acid in the lungs in the form of carbonic acid that's converted to CO2 and water, and then the CO2 is released when you exhale. So this shows you the arterial O2 content during these different pHs, and you can see as you have a decreased pH, your arterial O2 content goes down, and in the venous O2 goes way down, because those decreased pHs will greatly increase delivery of oxygen to tissues. And this is shown over here in the far side of the graph, 6.6 mils of oxygen per deciliter. And then during alkaline, you can see that your delivery is greatly reduced and your arterial oxygen levels become much higher. So all of these things are going to increase delivery of oxygen to the tissues, pH, increase temperature, and increase 2,3-diphosphoglycerate. They will so-called shift the position of the curve of the oxygen hemoglobin dissociation curve to the right. Okay, so we have some opponents of acidosis. Acidosis can be metabolic, or it can be respiratory. So if you don't have proper ventilation, as in cases of chronic obstructive pulmonary disease or asthma, not getting rid of enough carbon dioxide. If you don't get rid of enough carbon dioxide, then you're going to be generating more carbonic acid and will cause respiratory acidosis. Metabolic acidosis is usually due to the production of organic acids, lactic acid, ketone bodies, which are beta keto acids, things like that. Both of these will increase ventilation rates as a result, essentially, of the peripheral chemoreceptors. Alkalosis can be either due to hyperventilation, So you can have respiratory alkalosis, or metabolic alkalosis is usually due to excessive amounts of vomiting. That vomiting causes you to lose acid because now you have to produce more acid. That causes metabolic acidosis. Excessive steroid administration can also affect the blood pH as well. Let me just very quickly show you essentially why. So if you are hyperventilating, let's see, we can get this a little bit closer here. That's a little bit better. Remember, carbonic anhydrase, that's the enzyme that's really all over your body. This produces carbonic acid, and this disjoins to hydrogen ion and bicarbonate ion. If you are hyperventilating, that means you're getting rid of more CO2. Remember from chemistry, Le Chatelier's principle, that means you're going to drive this in this direction. Therefore, you will be losing more acid. Because you're losing more acid with hyperventilation, that's going to cause alkalosis. Whereas if you don't breathe, you don't have proper ventilation, this now goes up. That's going to generate more acid because you'll then, in those cases, push things this direction. Now that will create more acid. Very good. So now there are various genetic effects that can affect hemoglobin. Some affect the amount of hemoglobin, some affect quality of hemoglobin. So sickle cell anemia is due to a single base change. At position number 6, changes a glutamic acid to a vein. It provides a hydrophobic surface for hemoglobin. That hydrophobic surface causes hemoglobin now to polymerize under low oxygen conditions. This will distend the red blood cell, and these distended sickle-shaped red blood cells can clog vessels and cause tissue ischemia and infarctions and things like that. So here essentially is the sickling reaction of these erythrocytes. Incidentally, this disease is not called sickle cell anemia anymore. It's called sickle disease. It's not called that anymore. Okay, so it gives the sickling of the erythrocytes. Thalassemias can affect the quantity of hemoglobin that's produced. And these are usually due to mutations that affect the expression of hemoglobin. You can have alpha or beta thalassemias. Again, these are predominant in people of Mediterranean descent. These also tend to cause anemia. These are treated with a drug called hydroxyurea. Hydroxyurea will cause the expression of the full chain of the beta chain of hemoglobin, and that will essentially, it's not perfect, but it's better than the disease form that either doesn't produce any hemoglobin, or in the case of the sickle disease, doesn't produce functional hemoglobin. And that's how you get extra fetal globin fiction, is by treating with hydroxyurea. There's also a hemoglobin-like protein, but it's only one subunit, not 4 subunits, called myoglobin. It's found in it's found in muscle, particularly skeletal muscle. It has a much higher affinity for oxygen than does hemoglobin. It acts as a storage molecule. This does not show cooperativity. This will only release its oxygen when you have very, very low levels of oxygen in the muscle. So it's good as a storage molecule, not as a carrier. You can see this in the oxygen myoglobin dissociation curve. Even at very, very low levels of oxygen, it still will bind to its oxygen very jealously. Only when levels fall way, way, way below these levels here will it start to give up its oxygen. We've talked a bit about adaptation during exercise. So during exercise, you have increased muscle workload and increased oxygen consumption. You also have increased CO2 and lactic acid production. Therefore, you have increased oxygen demand. You have hyperventilation. Hyperventilation will blow off more CO2, potentially causing respiratory alkalosis. Exercise-induced ventilation, interestingly, is not prompted by alterations in gases. Instead, ventilation will increase abruptly as exercise begins due to neural effects. So first of all, there's conscious exercise anticipation. This is a psychological stimulus. You also have simultaneous cortical muscle activation of skeletal muscle. This will activate the respiratory centers, which are voluntary. Proprioceptors, moving muscles in the joints and tendons, they will stimulate respiratory centers. And then you'll have early compensation to exercise as opposed to waiting for change as you get change in blood values. Then you have abrupt increases in ventilation, which are followed by gradual increases, which will be reflective of lung CO2 delivery rates. You eventually get a steady state of respiration that's appropriate for the intensity that's achieved. And when exercise ceases, you have initial small abrupt decline in ventilation, followed by a gradual decrease in pre-exercise respiratory rate as you get a gradual decrease in carbon dioxide flow to the lungs. You have increased Bohr effect during exercise. You also have a decrease in pulmonary resistance because of the vasodilation. So you have perfusion of more pulmonary capillary beds. You have decreased physiological dead space in the lungs during exercise, and therefore increased CO2 delivery. Increased lactic acid production also increases the Bohr effect. Well, PCO2 and PO2 don't change. Arterial PCO2, PO2 don't change very much during exercise. PCO2 and PO2 on the venous end change significantly. And these essentially are partially responsible for the ventilation changes during exercise. A lot of your ventilatory increases are due to changes in pH, due to the Bohr effect, and due to neural effects as well. And these are summed up here essentially in this table. Increased ventilation during exercise is called hypernia. Hypernia. Hypernia is trying to maintain homeostasis by keeping oxygen and CO2 levels relatively constant. Hyperventilation is used essentially to blow off more CO2 that's been delivered. Once the muscle, striated muscle, reaches a level where it requires more oxygen than the blood can deliver, it now starts to undergo lactic acid fermentation. That point is called the lactate threshold. And endurance trained athletes with more mitochondria per muscle can go longer before the lactate threshold is reached. While pulmonary changes at high altitudes, there are fast and slow adaptations to high altitudes. Some of the fast adaptations occur immediately. Others occur over days. So first thing you get is increased ventilation because you have decreased arterial CO2. That increases respiratory alkalosis and increases, because you have increased blood pH, the chemoreceptors, peripheral chemoreceptors will offset increase in pH with an increase in ventilation rate. It's an initial effect. As adaptation occurs, the excretion of bicarbonate will increase. As bicarbonate concentration increases in the cerebrospinal fluid, respiratory alkalosis will result because of rapid ascent to high altitude. You'll have increased carbonic anhydrase inhibition. With increased bicarbonate excretion, you'll have mild compensatory metabolic acidosis, very slight. You'll also have an increase in 2,3-bisphosphoglycerate. That increases unloading of oxygen in the tissues. You'll have increased cardiac heart rate. For days and months, you'll eventually get right heart hypertrophy. That will give you increase. If you have increased pulmonary vascular vasoconstriction, you have increased pulmonary vascular resistance, or you get right heart hypertrophy, and non-essential functions therefore will be suppressed. You'll have more vasoconstriction in non-essential areas. Your later compensatory mechanisms include an increase in the production of red blood cells. That's called polycythemia. So these will make more erythropoietin. You'll have red blood cell made by the bone marrow. This gives you greater blood flow resistance. You'll have therefore greater heart workload. You'll have an increase in red blood cells, increase in hemoglobin, and an increase in the hematocrit. And this shows you essentially the changes as you get to higher levels. If you fly in a plane and you fly above 10,000 feet, it's recommended, what it's required that you use oxygen, essentially, because your saturation will now fall below recommended levels. And this summarizes some of those changes that we already talked about. If you go up altitudes too quickly, you can have so-called acute mountain sickness, otherwise known as altitude sickness. So the low atmospheric pressure causes low O2 levels. That causes hypoxia. This happened to my daughter. My sister took her for a hike. They went from 6,000 to 10,000 feet in a matter of hours, and Rachel got very sick. She started to upchuck, and they called Mountain Rescue to give her an IV. And it was due to the fact she ascended so quickly, 2,000 feet. So you supplemental oxygen, and if you upchuck, you just need to get electrolytes into you. All right. Any questions about the respiratory system?
I have one just about the last part that you were just talking about. Does that have anything to do with?
Andrew, back off your microphone. You're distorting and it's very hard to hear you.
Oh, how's that? That better?
That's a little bit better, yeah.
Does that have to do, is it a similar concept with like the bends when people are scuba diving and ascending?
No, with bends, you have increased pressure, so that with the bends, you have increased nitrogen solubility in the blood, and that's what causes the bends. The bends are due to the release of nitrogen, and that's why if you ascend, if you're scuba diving, you're supposed to ascend slowly, blowing out the air as you ascend, and that essentially will cause the nitrogen that's now starting to come out of your blood to get exhaled. That way you don't have all this nitrogen. That's due to increased pressure, not decreased pressure. So that's her mechanism.
Got it.
Okay. If there are no more questions, let's move on now to the digestive system. So digestion refers to the breakdown of complex molecules, smaller molecules that can then absorb. And that breakdown of small molecules into larger molecules into small molecules and absorption by our digestive system, that whole process refers to the enterprise of digestion. The digestive tract is divided into the gastrointestinal tract, which forms a tube that goes from our mouth to our anus, and also accessory digestive organs that attach to the gastrointestinal tract and are connected by ductal systems, usually exocrine types of systems, the liver, the pancreas, the gallbladder, things like that. So the functions of the digestive tract consist of, first of all, motility, moving the food from your mouth through the GI tract to the excretion. So you have, first of all, take food into the mouth, chewing it, that's mastication. So ingestion, taking food in the mouth, mastication, chewing it, deglutition, that's swallowing, and then peristalsis, whereby move the food bolus through the GI system from one end to the other. So motility. Second, secretion. You secrete exocrine secretions into the bolus that will help digest it, and then endocrine secretions that are secreted into the blood to regulate the digestive process and other processes that are linked to digestion. Then digestion refers to the breakdown of larger food molecules into smaller molecules, which then can be absorbed. That would be number 4. So motility, secretion, digestion, absorption. Throughout the GI tract is composed of 4 different layers. These 4 layers have different appearances depending upon where you are in the GI tract, but all 4 layers are there. These layers are known as tunics. Let's start from the innermost and go to the outermost tract. The innermost tract is called the mucosa. Below the mucosa, you have the submucosa. Below the submucosa, you have the muscularis. And then the outermost layer is the serosa. In some parts of the digestive tract, the serosa is called the adventitia, depending upon where it's attached. We'll get to that when we get to the different parts of the GI tract. So this is a cutaway of the different layers of the GI tract: mucosa, submucosa, muscularis, serosa. Notice that the mucosa is folded into internal folds. This is very common throughout the tract. We'll talk about those so-called plica circularis. And then the muscularis is broken into an inner circular muscle and an outer longitudinal muscle. These 2 will be responsible for different types of movements through the GI tract and then the outer serosa. This shows you the layers essentially from inner to outer. The mucosa is the absorptive layer. The submucosa, which is very rich in nerves and blood vessels. The muscularis, which has plexi from the autonomic nervous system. And the serosa, which is largely connective tissue, but also has the blood supply as well. All right, the mucosa. The mucosa consists of a simple columnar epithelium that is absorptive and secretory and lies on a basement membrane. That basement membrane is called the lamina propria. The basement membrane is composed of connective tissue and it's filled with lymph nodes. Within the mucosa, you also have goblet cells that secrete mucus throughout the GI tract. Here is a picture of a mucosa. You have the nice, beautiful, long columnar epithelia. You also have goblet cells. You can see them here that secrete mucus. These cells are highly polarized. You have an apical and a basal level. At the basal level, you have blood and lymphatic vessels. So things that are absorbed will be absorbed at the apical side and released into the bloodstream at the basolateral side. Here's some personal shot of a goblet cell. The goblet cell releases mucus at the apical side. This coats the mucosa, and that mucus is important protective layer. It protects the mucosa against shear forces and against enzymes. Below the mucosa, you have a thin layer of smooth muscle called the muscularis mucosa. This is a support layer. It folds the mucosa into these larger folds of plica circularis in the GI tract. These plica circularis folds are responsible for increasing the absorptive area. And it also is the anatomical delimiter that distinguishes between the mucosa and the submucosa. So here is your mucosal layer. This is a slice from the intestine. Here in the blue bracket here, that is your muscularis mucosa, and below this is all serosa. And you can see here, you have some lymph nodes. These are some of the lymph nodes, essentially, that you have in the mucosa. Beautiful columnar epithelia with goblet cells. Below the mucosa, you have the submucosa, which is very highly vascularized. This is the layer that is going to supply the mucosa with its nutrients. The submucosa also contains glands and also nerve plexi as well. So here is a picture of the submucosae. You can see that the submucosae are pockmarked with blood vessels. When you see an area of the GI tract and you see the perfusion of vessels, that tells you it's submucosa. So the V stands for the villi into which the mucosae are organized. M stands for muscularis mucosae, the anatomical landmark between the mucosa and submucosa. All these areas with blood vessels, that's the submucosa. Below that is the muscularis, we'll talk about next. Below the submucosa, you have a basement membrane, and then below that, you have the muscularis. This is what's responsible for the motility part of the GI tract. These contractions are going to move the food bolus through the GI tract. They're all going to pulverize the food bolus and break it up. As the food bolus is softened with digestive enzymes. The inner layer of the muscularis externa is a circular layer. This circular layer provides a ring around the gastrointestinal tract, and it's responsible actually for these types of radial types of movements. So these will contract and these will propel the food bolus. These are coordinated so that you contract and relax, moving the food bolus this way, and then as the food bolus moves, these contract, relax ahead, and so on and so forth. The longitudinal, which is outside the inner circular muscle, these are going to be responsible for this thing called segmentation. Segmentation is going to be a wave-like movement that's going to even move the food bolus faster, the GI tract. That'll work in concert with the circular contractions to move the food bolus through the GI tract. You have a couple of nerve plexi, the myenteric, otherwise known as the Auerbach plexus, that's between the circular and longitudinal muscle. You can see the structure here in this picture. That pretty obviously doesn't look like a muscle. That is a nerve plexus. That's one of the major nerve supplies to the muscle, the GI tract, so the myenteric plexus. A close-up of the myenteric plexus contains fibers from both the sympathetic and parasympathetic divisions of the autonomic nervous system. And then you have the Meissner's plexus. I guess in German it would be Meissner's plexus, which provides autonomic supply to the muscularis mucosae, largely the outer longitudinal muscle. And outside that, you have the serosa, which provides a protective layer of connective tissue. And then outside this large gob of adipose tissue, areolar tissue, you actually have a layer of squamous epithelia as well. Most of the innervation for the upper, mid, and lower GI tract is provided by the vagus nerve. And these parasympathetic preganglionic fibers are very long. They synapse with postganglionic neurons in the myenteric mucosal or Meissner's plexus. These postganglionic fibers then leave the ganglia, innervate their smooth muscles. Those postganglionic fibers will be quite short, which is very typical of the parasympathetic nervous system. Fibers from the sympathetic nervous system reduce peristalsis. They reduce secretory activity. They stimulate contraction of sphincter muscles, and they'll tend to hold food in various places. That's one of the reasons why nervousness, stress can actually induce so-called stress constipation. So travel sometimes can cause travel-induced constipation. That's due to activation of the sympathetic nervous system. These postganglionic fibers are longer. They pass through the myenteric and submucosal the ganglia and innervate their targets, the smooth muscle. All right. That's it for today. We will pick this up on Friday. And so we'll stop right there. And I will see you all come Friday, same time, same place. See you all.
What's the quiz on?
Quiz will be over the respiratory system. So that's chapter 16. Chapter 16.
Okay, is it the questions from the back of the book, or is it going to be questions you make up?
Well, I'll try to fit the questions from the back of the chapter into the blackboard, but those aren't always very conducive, so I'll do my best. So it'll be a black quiz. You'll get to do it on your own and so on and so forth. Okay?
Okay.
Okay.